All right. Next part. I think maybe even the last part. Yeah. Well, maybe we'll do the antivirals as a separate part at the end. I don't know. Yeah. Yeah. Next part. Let's go. So next part is RNA capping. Now, we don't know for sure when RNA capping happens. It has to happen at some point after we have started making the virus, and it could happen any point up until the virus is done being synthesized. But it probably happens fairly early in the process, would be the guess. Yeah. But yeah, really, we don't know. So it's just a thing that happens, and so we're going to cover it as a unit because we know the parts and what they do. And so let's do this. Okay, to make a cap, it takes 4 enzymes, and each of the enzymes has its own job to do. So shown up here, these little gray triangles, let's imagine these are nucleotides of the messenger RNA. And at the end of it, let's assume this little purple dot is a phosphate, is like an extra phosphate. So it's like a triphosphate end. And we don't want a triphosphate end. Oh my goodness, no. So step one, there's going to be an enzyme called terminal phosphatase. And its job is to cut off the phosphate, the last phosphate at the end. So we're going to go from a triphosphate down to a diphosphate. Terminal phosphatase is found in non-structural protein 13, which is also the helicase. Yeah. And it's actually the part of the helicase that would normally use ATP to do helicase activity will also chew the last phosphate off of something, which is, if you think about it, very similar to breaking an ATP down into an ADP and a phosphate, which sounds very familiar, right? It's a lot of what enzymes do. So the coronavirus is using its enzyme to do this terminal phosphatase activity. Next, we have the guanylyl transferase. And so what it's going to do is attach a cap which is backwards. So the cap is going to be a G nucleotide, so a guanine. And the guanine is going to be attached with its 5' end hooked to the 5' end of the next nucleotide. So that is just, backwards and wrong, but that is exactly how RNA caps get attached. And so that's why I've drawn it as a triangle that's pointed the other way because it's backwards. Yeah. Step 3 is we're going to have an N7 methyltransferase, and it's going to put a methyl group on that backwards G in a particular position on nitrogen at position 7. Yeah, great. And then the last step is we're going to put a methyltransferase. We're going to use a methyltransferase to put another methyl group onto the nucleotide after the cap. So it's basically the first nucleotide after the cap, or you can think of it as the second nucleotide, or the first one that's facing in the right direction. Yeah, whatever you want to think of it as. It's that nucleotide, and so that's the second methyl group. And so, in your cell, when you want to make a messenger RNA, you better believe you have all 4 of these enzymes lined up there. I think they are in the nucleus. And they're going to go through, and 1, 2, 3, 4, they're going to do their jobs and make a perfect little messenger RNA that can then get spliced and come out through a nuclear pore, just like Mother Nature intended. All right. So, who's my capping crew? My capping crew are these things. So, we've got our NSP13, which is a helicase some days, and a cap, or capping, terminal phosphatase on other days. And it's actually kind of the same thing. If it can't unwind it, it'll just chew a phosphate off the end of it and call it good. We've got the NERAN, the nitavirus RDRP-associated nucleotidal transferase. Turns out that is also a pretty good cap guanylyl transferase. And so it is able to do the backwards G attachment. Boom, there we go. There's G, only it does it backwards, like so. That's better. All right, and and that's part of NSP12. It's one of the domains. It's actually toward the N terminus of NSP12. Next, in capping, we have step 3, which is going to be NSP14. Now, you originally met NSP14 as the exonuclease, and that's this green domain right here, or the middle domain, if green is not a thing that works for you. That domain is going to be the proofreading domain. And then this bluish domain at the end, that is the one that is actually the methyltransferase. So NSP14 has two distinct domains that do two totally separate jobs, actually at two slightly different stages of the replication process. So neat. Yeah, and the protein is just going to be sitting there right next to the polymerase the entire time, so that it can do proofreading. And it just so happens that it brings along a capping enzyme attached to it. So neat, cool. Then the last capping enzyme is going to be the 2'O methyltransferase, or I just write it like 2O. And that's the letter O, not a zero. Because it's attached to an oxygen, that's what it means. And yeah, it's an O methyltransferase. It takes S-adenosyl methionine, that's what that SAM thing is in there. And it's going to use that as the source of its little methane group, and it's going to stick it onto the cap. So there you go. Now, for NSP14 and NSP16. So for our step 1 methyltransferase and step 2 methyltransferase, or N7 and 2O methyltransferase, they both require a little protein called NSP10. And so you can see this sort of gold thing at the side. That is NSP10, and it's holding on to this to hold it in the right shape so that NSP16 can do its job. And if you look at the end of this red thing, or if red is not a thing for you, this thing that's at the end, that is also a NSP10 protein. And it is required for all of NSP14 to actually function as well. So yeah, NSP10, pretty useful, pretty useful. And yeah, so those 5 things are the capping crew, as far as we know. And they're all going to be associated with the rest of the replicase proteins, which is why we think this all happens in the same spot, and probably they're going to cap as soon as they are able to cap, which would mean at the very beginning of RNA synthesis or after it's just started but hasn't gone very far. This is most likely. All right, let us go. So that was capping. Yeah. Now let's look at 3 little antivirals. And I think these are useful because of the way that they work. They're also useful in that they keep people from dying. Yeah, but useful from our point of view, like trying to understand the virus. So the first one is called remdesivir, and that was actually, I think, the first drug properly approved, or at least for emergency use approval, for coronavirus therapeutic. Yeah, so it's kind of neat. Yeah, there's some antibodies that were going through the process around the same time, but they're not really drugs. It's like a protein from somebody else that you put in, and you hope it does its job. And these are a little neater. So remdesivir is, if you look at that, can you tell? Is that a chain terminator, or is that something that gets put into the middle of the chain? Which nucleotide would you look at? Or which part of the protein, which part of this thing would you look at? So it's a nucleoside in that it is similar to a nucleotide. And so here's the actual nucleotide that it's most similar to. You can see there's the side. And really our only difference is that we put these little groups onto the side. And there is, look at that nitrogen triple bonded right over here. Those are the differences. And in the cell, we're actually going to shed off these 2 things. Those just are there to help it be delivered as effectively as possible. The cell is going to activate it just like it would activate any nucleoside that it has, and it's going to add phosphates onto it. And so instead of being going from AMP to ATP, it's going to go from remdesivir to remdesivir triphosphate. Yeah, why not? And it's the neatest thing. It's not a chain terminator, but it's a 3-step chain terminator. So I don't know if you've seen any old kung fu movies or like Kill Bill or something like that, but there are these ideas going around in ancient martial arts films of a secret punch that is so devastating that you'll take 1, 2, 3 steps after you're hit, and then you just die. Like like your heart will explode. Yeah, amazing. Remdesivir is a 3-step killer, 3-step punch. So step 1, remdesivir is going to be phosphorylated to make the triphosphate. Step 2, remdesivir is going to be incorporated by the unsuspecting viral polymerase, because the polymerase is not all that careful or all that smart. Step 3, the virus is going to keep on putting things in after it. And that's fine, because remdesivir would make a total mess. If you went and tried to base pair something with this, I guess you could base pair a T with that probably fairly well, but I don't know, this azide, or whatever that is, group on the side, that's a little weird. But then what happens is you go 1 step, 2 step, and then when you get 3 steps away, or 3 steps away up here, that's where the virus just stops. And the polymerase gets jammed. And so I don't know if you noticed, but let's go back and look. NSP8 actually does 2 things. So it's that primase that sits up front, and you see these long parts there like an exit tunnel. So this twisty thing in the center, that's actually the viral RNA. And the red spot here, that is us putting on a new nucleotide, a viral RNA. So pretty cool. And with the extra little group here, it just sticks out just barely far enough that it blocks the exit tunnel, we think, and just ends up stopping the polymerase. The polymerase can't go any further. So it goes 1, 2, 3 bases, and that's where it runs into the side, and it just gets locked in place. And then the polymerase is dead and is stuck to that particular nucleotide. And eventually it's just going to break down, but it's not going to make any copies. So good job, remdesivir. Way to go. Yeah. Second, antiviral, molnupiravir. And so molnupiravir is another one that is going to be under emergency use authorization. And molnupiravir, again, have a look at it. So here's a molnupiravir, and here is the C nucleotide. And you can say, okay, so what do we? Actually, no, that's not a C nucleotide. It's got an extra little hydroxyl up here. Otherwise, it's very similar. But yeah, it's just going to get phosphorylated, and instead of matching perfectly like a C would to a G, instead it'll also match fairly well to A and a little bit to U and pretty darn well to G. Yeah, as if it was a G. Yeah, so. And it has a great effect on the virus. It really controls replication and seems to make people better. So that's fantastic. So the way it works is by pushing the virus into error catastrophe. So the virus is on its little peak of fitness. It's sitting right up there at the top saying, you can't touch me. I'm SARS coronavirus 2. And what this does is it goes in there and the virus puts one in, maybe puts another one in, and every step it goes boom, boom, down the hill. Because once you put in a molnupiravir, every time the virus goes back to copy that strand, whether it's in this cell or whether it gets packaged and sent to the next cell, it's got to put in a random nucleotide at that position. And random mutations are not good for anybody. Yeah, that's the Fukushima rule. Yeah, it's just, radiation does not do anything good for people. It only does bad things for people in general. Yeah. All right. Drug number 3, because why not? Let's talk about a Texas A&M thing. So this is one of several sort of competing main protease inhibitors that are out there. And this one's called MPI-8, and I included it over some of the others because this one has a name that's short enough and memorable enough that you can actually remember it. And because it was developed right here at Texas A&M University in the Liu lab. Yeah. He's a very smart guy over in chemistry. And so the way it works is it just goes in. It looks like a peptide. It is what we call a peptidomimetic, PEPTIDOMIMETIC, which just means a thing that mimics a peptide, which this does. And then it has some extra little funky groups sitting off at the side to help it sort of bind a little bit better to the active site of the main protease. And yeah, it does a good job. Does a very good job of that. So there you go. And so if you inhibit the main protease, then you can't cleave NSP 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16. And if you can't cleave those, then the virus is dead. So that's how it works. Yeah. All right. Hope you got something out of that. Hope you liked it. Yeah. Hope you understood it mostly. That's what we're here for. All right.